Hello. Transcription, once initiated, will keep on happening, and that is elongation. Not much happens during elongation in terms of variation except for the proofreading. But should transcription keep on happening, or should it stop? If it keeps on happening, then you are unnecessarily transcribing a lot of DNA that is not required. It's a waste of resources. At the same time, if we stop abruptly, we might not be producing enough of the information for a gene to produce protein. So transcription should happen in a very orchestrated or systematic way so that there is no wastage of resources, and at the same time there is expression of the gene, and that process is called as termination. In prokaryotes, the terminators, what you call them, are present in the ends. Say for example, if this is the promoter, we have an open reading frame, and at the end we have a terminator, so that only that part of the gene or the genome is transcribed, and there is no wastage of any resources in terms of nutrition or the matter, that is the bases should be added, and energy required to make these bases. And not only that, when we degrade, the RNA should be degraded at the end, and to degrade again we have to spend energy. So terminators become important, or termination it becomes an important step as well. In bacteria, there are two different types of terminators that are present. One of it is called as the row independent terminator, and the other one is called as row dependent. It is basically based on one factor called as row protein. If a terminator requires that row protein, you call it as row dependent, otherwise not. We will look at row independent terminator, and this is also called as intrinsic, because when you say intrinsic, it is within itself. That means when transcription should happen is already encoded in the genome itself or in the DNA. How is that possible? It's interesting to note that the terminators usually have some kind of symmetry, dyad symmetry. That means if you see this region is complementary to this region. Say for example, this is the DNA that is present, and here is the RNA that is produced here. The underlined one are the regions that are complementary. So if they are complementary and the produced RNA is single-stranded, so it can fold upon itself and start base pair. Although we say what are the differences between DNA and RNA, we often say RNA is single-stranded. It can be single or double. And because there is no complementary strand, and there is a propensity for the bases to form hydrogen bonds, wherever there is complementarity, they would fold upon themselves. So RNA form complex three-dimensional structures, and sometimes are useful or involved in regulation of many things. And in this case, the terminator could form a hairpin loop like this, and that loop. And there is also one more part of it, is that there are stretch of U's, and when a structure like this forms, say for example, this is where the RNA polymerase is at this point, and a structure like this has formed, that kind of signals or changes conformational change of RNA polymerase, because of which termination happens. So that is why they are called as row independent, because there is no involvement of a protein called row. And more appropriate name is it's an intrinsic terminator, because the ability to terminate the transcription is encoded in the way the design of the nucleotide sequence here. It is within the sequence itself, and that's why it's an interesting and nice way of seeing termination. The other one is called as the row dependent, because it is there is a protein called as row protein, which has affinity for regions called as RUT. So assume that this is the gene encoding, and at the end of the genes, the 3' side of the open reading frames or those, we may have something called as rut sites. Sequences vary from species to species. And if the RNA polymerase was transcribing it, the mRNA would have the rut site, and that is where Rho protein would bind. And that binding of Rho protein to the rut site in the RNA being transcribed, or the RNA polymerase is stimulated to terminate the transcription, and that is why we call it as Rho dependent terminators. So termination occurs is an important step. There are two things. First thing, it should happen at specified loci or locus, because we don't want any unnecessary transcription. At the same time, we don't want shortage or formation of short RNAs or incomplete RNAs. And there are two types, Rho independent and Rho dependent, based on whether Rho protein is required or not. Rho independent ones are intrinsic because they can form the sequence in such a way that they can form a hairpin loop, and that can stimulate transcription termination. Thank you very much. See you later.